Hi, my name is Christina Waters, and I am the CEO and founder of Rare Science. I'm also the Senior Vice President and General Manager of the Global Rare Disease Program at WuXi NextCode. I was speaking today about accelerating cures for rare childhood diseases by building global communities and inducible pluripotent stem cell collections through my nonprofit, Rare Science. Rare Science is a nonprofit research organization with the mission to accelerate finding more immediate therapies for kids around the world with rare disease. And it is an honor, and I thank the organizers for allowing me to be a part of 24-hour Stem Cell Day. The challenges of rare disease. Even though rare disease is fundamentally rare, it actually affects 300 to 400 million people around the world. More than half of these affected are children. So that means that we're looking at an estimated 200 million kids around the world that have rare diseases. These patient populations are very small, and we've identified about 8,000 different rare diseases currently. Rare diseases are responsible for 35% of the deaths within the first year of life. And from this group, 30% of these children will die before their 5th birthday. And this is at odds with the current drug development process, as it takes an average of 14 years and $2 billion to develop a new therapeutic. So we need to find another solution to address the urgency that is needed for kids with rare disease. The challenge is also that we only have FDA-approved treatments for about 5% of the different rare diseases that have been identified. Many of these rare diseases look the same, and so it's very hard to diagnose them. On average, it takes 7 years in the US and 5 years in the UK for a correct diagnosis. And during this time, these kids could be misdiagnosed 2 to 3 times and see 4 different physicians and specialists. And during this time, their disease is actually progressing. So in the rare disease patient journey, it's very different than what we're doing in rare research and development, which also causes some challenges. From the patient side, we're looking at parents that have an urgency to find a diagnosis for their child. They need access to information. They're trying to understand if there are available therapies that could be used for their child for improving quality of care and even a cure. Where do they go for support and guidance? And then also access to these therapies, even if they are available, is difficult. And then also the cost of these rare disease therapies are very expensive. And then on the other side, we have research and development, where they're trying to understand how to get access to patients and biosamples that are scattered around the world, in addition to the data that is also located in various places around the globe. These data sets are really small. It's very hard to get statistical significance on understanding the biology, the causal underpinnings of these diseases. The research and clinical strategies are not often coordinated. And clinical trials in the regulatory process, even though we're making great headway, aren't always adopted for the use of a clinical trial that could help with such a small patient population. And also there's the challenges to access to funding. So even though the cost of DNA sequencing is going down, still we don't have a lot of access, or patient families don't have a lot of access to getting their child sequenced when they are going into the hospital with some type of unknown cause. So we have also an understanding that currently 30% of the time do we actually identify a known gene or gene variant that's causal. And as I had mentioned before, even if we do identify the genetic etiology of these rare diseases, there's only 5% of the time there's a therapy that's available. So even with identification of a causal gene, the other complication is that we don't always understand how the gene or the gene variants are working in concert with other gene mutations in the body and how they're changing the biology that actually leads to clinical phenotypes that we can treat. So on this next slide, it's very busy, but I wanted to illustrate from the top down and the bottom up the importance of being able to translate the understanding of gene function and integrating it into the phenotypes that we see. So from the top down, in this example of neurology, we have a lot of different diseases that look the same. They have names like Fragile X, autism, SYNGAP, and ADCY5. All of these different diseases, rare diseases, actually have developmental delay, but only Fragile X, autism, and SYNGAP show cognitive delay and have parts, some of these kids have epilepsy. ADCY5, on the other hand, is a motion disorder and oftentimes misdiagnosed as cerebral palsy. But as you can see in the boxes below, under tissue specificity and expression of the protein, you can see there that the expression of these genes and the proteins that are associated to these diseases are expressed differently in different tissue types. And so it's so important to also, again, go from the bottom up, sequencing, understanding what genes are actually have mutations, what are the molecular pathways that are involved, and then again, looking at what tissues could potentially be affected and have a changed biology. So the value of inducible pluripotent stem cells is incredible in the way that we can utilize them to do a couple of different things for rare disease. On the right-hand side of this slide, you'll see that, and we've often talked about how currently in the news, we're thinking about how we can use stem cells as a therapy itself that are derived from the patients that are sick. On the left-hand side of this slide, it also shows how the inducible pluripotent stem cells can be used for disease modeling. So we can actually take tissue from a patient, create these inducible pluripotent stem cells, and then start looking at the function of these stem cells once they're derived into the tissue of question. There's the possibility of building functional assays, and from those functional assays, there is a hope that we might be able to even repurpose currently approved drugs or even identify new molecular targets for new drug development programs for rare disease. So I wanted to share an example of one of the Rare Sciences' first projects and how it works working directly with patient families and how we're hoping to understand the biology of the ADCY5 gene. So in the middle of the picture, I wanted to introduce the Grossmans, Steve and Gay and Lily, their daughter, who I met at a patient advocacy organization and conference. And Lily, their daughter, had been misdiagnosed for some time with cerebral palsy. However, she did get sequenced, and they identified that the gene mutation was in ADCY5. The challenge is that nobody understood how this ADCY5 gene contributed to such a severe motion disorder. And they also felt they were the only one in the world that had this gene mutation. And so what we did was work with ADCY5.org, an organization that was co-founded by both Gay and Steve, and identified families on the right-hand side of the slide that also had children that had ADCY5, the mutation. What we found was that there was a huge difference in the way that this gene affected the kids. There were kids that were very severe and in power chairs to seeing some of the kids that actually played soccer, but they all had this mutation in the gene. So how do we start understanding, again, what is the biology that's causing these diseases? How do we validate which variants or which mutations actually cause severity? How do we also identify the downstream biology or pathways that could be druggable, that could lead to a potential therapy to help these kids? So what we did know about ADCY5 pathophysiology is that it encodes a regulating synthesis of cyclic AMP. And so this family, which is, there's 10 different forms of ADCY, this number 5 is highly expressed in the striatal tissue of the brain, which is known to be responsible for motion, and then also in the myocardium. But again, there was no understanding how the gene mutation resulted into the clinical phenotype. As seen in the picture prior, we saw a wide spectrum of observable traits and different severity. We also had an understanding at the very early on with some of the great work that had been done by other scientists in this space that there was some mosaicism, different cells expressing this mutation than others. But what we did identify was that all of the kids that had a mutation in the ADCY5 gene had a mutation in the cytosolic regions of the protein. So it absolutely seemed to have some type of an effect on cyclic AMP. So what we wanted to do was actually start understanding, again, could we create a stem cell program and create stem cells and hopefully dopaminergic neurons to actually study the biology that was related to the motion disorder. So what we did was we worked on doing patient outreach, finding families around the world that wanted to participate. We did an IRB approved consent for these families. We also created an 18-page medical history survey understanding the phenotype of each of these kids, what drugs that they or therapies they were responding to, how severe they were, and again, really to start understanding what was the connection between the variant and the actual phenotype that we were seeing. At the bottom of the left-hand side, that graph, we actually identified 5 to 7 different variants, but again, they were all in the cytosolic loop of the protein. And then the world map actually shows all of the families that participated, which was 5 countries across the world and 10 different families and greater than 50 different biosamples that we brought back to be able to start building these stem cell collection. So you can imagine this is quite an undertaking, and a lot of great people were involved. My nonprofit, Rare Science, is a completely volunteer organization, and we work with all of these great companies that helped us with different expertise to be able to build this stem cell collection so that we could lower the barrier for researchers around the world to start, again, understanding what the biology was that was causing what we saw as this clinical phenotype. CIRM was instrumental at the California Institute of Regenerative Medicine, which was funded by state voters in California by Proposition 71 that authorized $3 billion of funding that went towards building out stem cell collections. We were one of the first rare disease stem cell collections that have been built through this effort. So as you can see, we had been working with CIRM, Cellular Dynamics, All Cells, Coriell for banking, Thermo Fisher Scientific was very helpful in organizing introductions and reagents, to working with Aspire IRB for our consents. For vials, BD, we also worked with hospitals around the world to help us collect the samples from the patients that were going to be sent back to Cellular Dynamics and All Cells for processing of the stem cells that we were making. So what we can do now to understand rare disease and potentially have this be a model for many of the other rare diseases that we understand what maybe the gene is, but not understanding the biology. We hope that this can demonstrate and model many other opportunities, and we can scale this concept. But now that we had the stem cells made from all of these different families, not only the affected child, but also normal siblings and parents, we're able to now start differentiating into dopaminergic neurons and other tissues that ADCY5 is highly expressed to really start understanding that biology. We can compare the differentiated tissue of the different variants of the different kids to see if there is a connection between the variant and also the severity of the disease. We can also use CRISPR to revert the mutations back to normal, in quotes, so that we can validate that this actual mutation is causal to the biology that we're seeing, and then also identify whether there's other contributing genes or mutations in that one individual's genome. In addition, as I mentioned, we had a lot of the kids' mosaic. And for some of this collection, we were lucky enough to be able to create clones that have the mutation of ADCY5 and also a clone that doesn't. So these individuals can serve as their own control and understanding how those different tissues are functioning differently. We can also create functional assays to test therapies directly using the individual's tissue. And we can also identify, hopefully, new molecular targets downstream that are affecting, again, the biology. And what's also now so fascinating is that we're not only able to start building out different tissue types, especially that are important for ADCY5, like the cardiomyocytes and the dopaminergic neurons, but we have also the potential of building and growing organoids, and hopefully we'll be able to look at mini brains to be able to start understanding how this could potentially be this mutation affecting development and also adult function within a three-dimensional organ. So what we're currently working on right now, and again, this is in partnership and collaboration with many different groups, and this is how we, again, hope to scale our program at Rare Science and offer this to more rare disease families, is that the University of Lübeck came forward, and after we started building our clones, they offered sequencing to ensure that we really did capture the mutation and what that was. We also have been, again, making these, we will expand these cells to P10s and make them available for collaboration for scientists around the world to be able to start looking and identifying, the biology and answering different questions that might be important. We also, through the University of Humboldt State, we have graduate students that are already starting to look at the role of cyclic AMP and the dopaminergic neurons, not only during differentiation or after differentiation, but before, seeing if there's any difference in the populations that occur with and without cyclic AMP. And we had CERM students from Amy Sproul's lab at Humboldt come down and also spend time with Jean Loring at Scripps to start understanding how these different mutations are responsible for different phenotypic characterization of the different dopaminergic neurons that were created. So you may be wondering how we actually find these families. And so currently, how we want to scale and build our program is with the 1,000 different rare diseases that we're currently working with. We have reached 5,000 kids, and this is across 50 countries. And we actually do this in a very unique way, and that is, as I was saying at the very beginning, these kids are scattered around the world. There's islands of data. How do you bring people together? And we do this through our unique program, which is the Rare Bear program, where we have people make one-of-a-kind bears for one-of-a-kind kids around the world, and we use social media to be able to explain and describe what these diseases are. Because again, a lot of these kids are misdiagnosed. The Rare Bear Army is consisting of about 5,000 members currently across 12 countries, every state in the U.S. We have the craft companies actually helping us sponsor the making of these bears, which the community coins as bear skins. The bear skins come back to San Diego, California, and we work with philanthropic fraternities, the Girl Scouts of America, Boy Scouts, biotech, and pharma. And you can see we've had a lot of sewing and stuff-ins of these bear skins right here in Carlsbad, California with Thermo Fisher and many of the other companies in our community. So again, we've really brought a grassroots community together and made everybody instrumental in identifying kids around the world with these rare diseases to help us understand what's different and what's similar. And from that, we will be able to continue to start characterizing and seeding new research with the hope to be able to understand the biology and build a faster, accelerated way through inducible pluripotent stem cell collections, new therapies that will help these kids. So as a takeaway, as we start really thinking about how sequencing is no longer really the challenge, we need to eliminate the barrier of access to sequencing to all families that need it. The challenge of rare and common disease will not be the interpretation and validate the sequencing of the DNA, but the challenge will be the interpretation and validation of causal genes, new ones, that 70% that we still don't understand why these kids are sick. How do we validate these variants? And this can be potentially overcome by using the iPSC cell line. Rare disease and new technologies such as the iPSCs can reveal, again, new biology and pathways that are masked by complexities of common disease. Empowering rare disease patient families with tools to build global communities and drive research to the clinic will impact global health. And then we can leverage what we learn from one small rare disease to diseases of similar biology, including common disease. And I believe that the best healthcare is the most cost-effective, and that's how, if we can marry the technologies that are available out there by accelerating, identifying the biology and helping these kids in the more immediate, before their disease progresses, we help everyone. And then lastly, I leave you that we can never underestimate the power of the sharing economy of good and what we can all do together through volunteering and also uniting all of our expertise in a new way and new pathways to help kids with rare disease. Thank you again for letting me be a part of this great day. And I leave you with our contact information. If you would like to reach out to volunteer or learn more about our program, you can email me at cwaters@rarescience.org for volunteering, info@rarescience.org. And our website, you can reach us at www.rarescience.org. If you would like to see the pictures of the bears and the kids they've been gifted to, you can also see that at the Rare Bear Kid Gallery on our website. Thank you again.